Okay, so let's start and good morning. Welcome to this new episode of the ESDR Kitchen. And this episode is a little bit different because we have the honor to have not a dermatologist or a PhD that does skin research. We have a rheumatologist, and it's a really pleasure for me to introduce Francesco Ciccia. We are colleagues because we work in the same university in Naples. He's the head of the Department of Rheumatology. And I'm very, very happy to have this episode because as the SDR wants to highlight skin research, we all know that skin is not just an envelope. It is connected with the whole body. So the joint, we know no, that they are well connected with the skin, and psoriasis is an example. So we are very curious to hear the journey of Francesco in doing research in psoriatic arthritis. Just a little information before starting. I want to say all the participants that you can make questions in the chat or in the Q&A function, and we will discuss at the end when the talk will be finished. Thank you. The stage is yours, Francesco. You're muted. The microphone. Francesco?
Okay, now can you? Okay. Sorry. Thank you, Anna, for first of all for the kind invitation. It's a pleasure and honor for me to be here in this nice meeting, and I think it's a very, very interesting opportunity, and it's a very good model, and I think we can also try to export in our rheumatological setting. I'm an intruder because I am a rheumatologist. As Anna said, I work the University of Campania, Region of Italy in Naples, and we have very close collaboration with Anna, just not only sharing patients, but also sharing ideas regarding the pathogenesis of psoriatic arthritis, and obviously working with two different points of views, because obviously we just treat sometimes the same patients, but a different perspective. Let me start just with the first slide that you perfectly know is nothing new, but this is just the perspective of a rheumatologist. The idea of the pathogenesis of psoriatic arthritis is that in genetically predisposed subjects, in this case, we have just put a slide that is more related to patients with axial disease. So in genetically predisposed subjects, there are alterations in the composition of microbiome, not only in the context of the gut, but also in the context of the skin. We're talking about psoriatic arthritis. And this is responsible for the activation of mainly innate immune cells that belong to the branch of type 3 immunity. And these cells, by producing IL-23, by producing IL-17, but also TNF-alpha and IL-22 are responsible for the different manifestation of the disease. One point which is absolutely important in our mind, just in the mind of rheumatologists, that the disease is highly heterogeneous. So we have a different clinical manifestation with different phenotype of disease that we are not completely sure that are just related to the same immune pathway, to the activation of the same pathways. And this is something that we are trying just to address in our research, just try to see whether it is possible to have a more personalized medicine in our patients, try to stratify our patients according to the different phenotype, trying to understand whether different phenotypes are just followed by different pathotype. But just talking about this journey in the pathogenesis of the disease, in our ideas about the pathogenesis of the disease, I have to tell you that we have a lot of controversial. One of the first controversial is related to the possibility that what we have learned in the past 20, 30 years, possibly more than 50 years, from the concept of spondyloarthritis by Mullen-Wright, is that axial spondyloarthritis and peripheral spondyloarthritis essentially are the same from a pathophysiological point of view. What we now are trying to try to demonstrate is that the two diseases are possibly similar at the beginning of the process, possibly the mechanical the mechanical stress, the biomechanical stress that is transduced by the enthesis to the site, to the attachment sites, is important in both the disease, but possibly in axial spondyloarthritis, we have a more inflamed bone marrow and activation of innate and adaptive immune cells that reside in the context of the bone marrow, and this is responsible for the main manifestation that we can see in our patients at imaging, which is the bone marrow edema. And it is something that also happens in normal subjects, but in patients with axial SPA, for the genetic predisposition, for the presence of altered microbiome, this is a chronic inflammatory response that just leads to the clinical manifestation that we can see in our patients. What now we are thinking about psoriatic arthritis is that this is not just a myelopathy, such as axial SPA, but more a pure enthesial inflammatory disease. And you can see in this cartoon from Dennis McGonigal that the classic parasyndesmophytes that we can observe in our patients with axial disease is essentially related to the inflammation of the attachment of enthesis to the bone and not inflammation of bone as a primary movement. This is something that is important because can help us in just try to think in a different way to a different condition and try to imagine that the two conditions are not the same from a pathophysiological point of view. And just an example, this difference is related to the different response to IL-23 inhibitors that absolutely work in patients with psoriatic arthritis and absolutely do not work in patients with axial SPA. So this is something that might help just the heterogeneity of the disease at the site of the primary target organ. Another problem in our mind, and this is a problem that we share with you, is the possibility to intercept patients with a very early phase of the disease, patients with preclinical psoriatic arthritis. Different classification, different ideas have been published regarding the stratification of patients according to the different disease phases. What we think, and this review point recently published on Lancet Rheumatology, what we propose is that the only preclinical phase, the very preclinical phase of patients is the phase in which patients do have psoriasis, but they do not have any kind of signs or symptoms of joint inflammation or enthesial inflammation or spinal inflammation. This is the true phase, the true preclinical phase, in which possibly you can intervene by using biological drugs, and by using these drugs, possibly you have the possibility to modify the natural history of the disease, the natural progression of the disease from psoriasis to psoriatic arthritis, at least in a percentage of patients. The other phases of the disease possibly are more early phase of the disease. So if we observe a patient with arthralgia, without arthritis, but with the evidence of enthesitis or synovitis, possibly these are patients with a very early disease, such as the patients with arthralgia or clinical evidence of enthesitis or synovitis that lasts more than 1 or 2 years. This, I think, is a very important consideration because just a split in our just conceptual view of the problem, because the only phase in which we can intervene just for modifying the disease is this phase, is your duty. But the problem is that possibly this is true only for patients that are coming to your outpatients clinic, not for our patients, because we are continuing to observe patients with mild psoriasis, very few manifestations of skin disease, and our patients have more joint or enthesial inflammation. So possibly this is the concept of just interception of the transition between psoriasis and psoriatic arthritis is something that is more relevant for the dermatologists, less relevant for rheumatologists, but is something that need to be better addressed in the next future. The third part of the third problem that I have in my mind is the pathogenesis of the disease, because we really do not know nothing about it. We really know we have very few information about the pathogenesis of spondyloarthritis. We are continuing to ask ourselves whether or not spondyloarthritis are more autoimmune or more autoinflammatory disease. This is a crucial point because we are continuing to assume that type 3 immunity, IL-23, IL-17, are the key cytokines, the hierarchically more important cytokines involved in the pathogenesis of the disease. But we have more than 40% of patients that do not respond to drugs that specifically target IL-17 or IL-23. So possibly we have a more heterogeneous disease, we have a higher heterogeneity, and possibly we can just start to think in a different way. Possibly some patients have a more autoinflammatory disease, some patients have a more autoimmune disease. The majority of the patients possibly are just in the middle of this just theoretical scale that I'm showing you in this cartoon on the right part of the slide. But one of the most important, the two most important part, not the most important, but the two relevant part of these stories are represented essentially by the genetic predisposition and by the possibility that different compartments of the body of our patients, the skin or the gut, are colonized by just altered bacteria, are colonized by dysbiotic microbiome. This, I think, is a very important part of the story. It's something that absolutely requires our attention because we are just at the beginning of a very story that might modify in the future our clinical approach to our patients. We have different data, published data in the dermatological field, highlighting the presence of dysbiosis in patients with spondyloarthritis. You can see here listed different papers from different groups that have evaluated people coming from different countries with the analysis of different samples and different techniques. What I can tell you is that in all the papers that have been published, there is a dysbiosis in patients compared to the controls. However, we cannot compare the studies because the studies are completely different. So we can compare apple with peach, for instance, because we are not talking the same language. And this is something we are working on, trying to homogenize not only the sample collection, but also the analysis of the samples and the interpretation of the results according to different factors. And this is something that I think is absolutely important in improving our knowledge of the dysbiotic, the microbiome world of our patients. Obviously, we don't know exactly what came first, and we don't know whether dysbiosis is a first moment of the disease or whether it's a consequence of the inflammation, is a consequence of the systemic inflammatory process that occurs in our patient. This, I think, is a very crucial point because if the dysbiosis is something that just precedes the onset of the inflammatory manifestation is something that we can address in some way in our patients, possibly also preventing the onset of the disease. Obviously, we have just to precisely identify the phase in which we can intervene in our patients. Otherwise, it's quite complicated to imagine the possibility to modify dysbiosis once this is just established after the onset of inflammation. In this, this is a very big problem, and possibly some data coming from the axial SPA, the HLA-B27 related disease, might help us in trying to give an answer to this question. You know that HLA-B27 is strongly related to axial spondyloarthritis. It's present in 35–40% of patients with axial psoriatic arthritis. It's the strongest genetic association in medicine between genotype and phenotype. this absolutely has been a very intriguing field of research for rheumatologists. And what we have just learned and studied when I was student was that the possibility that HLA-B27 just predisposed to spondyloarthritis by presenting self-antigens, autoantigens, cross-reactive with just foreign antigens to immune cells, activating the innate or adaptive immune response in our body. This is the arthritogenic theory. And this has been demonstrated. The relevance of this theory has been demonstrated 3 years ago in this paper published in Nature, in which the authors took peripheral blood, synovial fluids, and uveal fluids from patients with HLA-B27 related disease. They analyzed the peripheral blood of these patients by single-cell RNA sequencing, and what they observed was the presence of clonotypes of CD8 positive T cells in the different target organs of these patients. So, in other words, there is the innate part of the immune system is activated, but with a clonal expansion of cells that are possibly autoreactive against some type of autoantigens. And the same group, the last year, published just an update of the study. What they found was essentially deeper analysis of cells isolated from uveal fluids and synovial fluids of patients with HLA-B27 related disease. They confirmed the presence of autoreactive CD8 positive T cells, but interestingly, what they found, what demonstrated was that these cells were derived from the gut. These cells were alpha-4 beta-7 positive, which is a marker of intestinal homing. And so in other words, there is something that is happening in the gut of our patients, and is able just to expand, activate immune cells that are responsible for the inflammatory process. And when the authors tried to identify the autoantigens that were recognized by these cells, they discovered different autoantigens, JAK3 for instance, but many of these proteins were proteins that were related to the neurotransmission, to the nervous system, which is something of really interesting also because many years ago, 25 years ago, an Italian researcher, Maria Teresa Fiorillo, working in Rome, already demonstrated the presence of clonotype of T cells in patients with spondyloarthritis. And what she just showed us was that T cells were autoreactive against the receptor type 1 of the vasoactive intestinal peptide that was structurally just showed a structural homology with the latent membrane protein 2 of the Epstein-Barr virus. So there is something happening in our patients, possibly in the gut, which is related to the transmission of the VIP signal in the gut. And why VIP is so important? It's important for 2 main reasons. The first one is related to the ability of VIP in controlling the fucosylation of epithelial cells. This is just a modification of the surface of the epithelial layer, and it essentially consists in the activation of this enzyme, FUT2, which is responsible for the generation of fucosyl conjugates that are just secreted by the epithelial cells, forming just a protective layer that is essential for the maintenance of eubiosis, because these fucosyl conjugates are just the food for good bacteria, for instance, Bifidobacterium, and this process is controlled by VIP. When the vipergic fibers are destroyed, for example, by just giving alcohol to the mice, there is no fucosylation of epithelial cells, and this is sufficient by itself for inducing this biosis, for inducing the expansion of bad bacteria. So you can imagine how this might be important, because this might be just the explanation of what came first. It might be absolutely relevant in our story. And the second reason why VIP is important in our journey is that VIP is a very anti-inflammatory protein. This is an old paper published 24 years ago, in which the authors tried to treat mice with arthritis with an agonist of VIP. And what they displayed was that treatment with VIP agonist was able to significantly reduce the inflammation in these mice. So VIP is an anti-inflammatory protein, which is important in controlling, in modulating inflammation in mice. What about humans? I'm showing you just some results coming from my lab in which we have analyzed the expression of VIP and the expression of CD8 in the gut of normal subjects. And what you can see here is that there is the presence of Vipergic fibers in red that are surrounding intestinal crypts. In green, you can see CD8 positive T cells. And in higher magnification, you can see that there is very close interaction between the Vipergic fibers and CD8 positive T cells, also in the gut of normal subjects. What happened in the gut of patients with spondyloarthritis? There is a disruption of the expression of VIP. You cannot see Vipergic fibers. The length of fibers is significantly reduced, while there is a very significant dramatic increase in the number of CD8 positive T cells. This is confirmed at transcript levels by real-time PCR, especially in the male patients, and especially in the patients with higher degree of intestinal inflammation. And according to these results, also the expression of FUC2, which is the enzyme regulating the fucosylation of epithelial cells, the expression is very dramatically reduced in patients with axial SPA, and this is accompanied. You can see a difference in fucosylation of epithelial cells in red, and you can see that fucosylation of epithelial cells in patients with spondyloarthritis is dramatically reduced. And this is a process that we can observe also in the synovial tissues of our patients. You can see that, especially in patients with the lymphoid pathotype, in the more inflamed synovial tissues, the expression of VIP receptor is dramatically reduced. So this is our hypothesis we are working on, essentially in genetically predisposed subjects. Autoreactive CD8 positive T cells might induce the depletion of Vipergic fibers. These are responsible for the onset of dysbiosis, these are responsible for the activation of immune cells in the gut, but also responsible for a reduced VIP levels in the context of the joints. And this might be responsible for the inflammation that we can observe in our patients. Obviously, the next step is trying to demonstrate that this idea is mechanistically relevant, and what you're trying to do is just treat SKG mice, which is a model, they are mice, a murine model of spondyloarthritis with VIP agonists, to see whether we can just prevent the onset of spondyloarthritis. The last 2 minutes of my presentation will be just related to the possibility to just not considering close the therapeutic story of our patients, because actually we have different drugs that we can use in our patients, blocking IL-17, tumor necrosis factor alpha, JAK inhibitors, IL-23, and so on. But we have still just a very high percentage of patients that are not responding to this compound, and possibly we need new therapeutic targets to be addressed in our patients. We are working the just also on the possibility to modulate the epigenetically modulation of the transcription of pro-inflammatory cytokines. And one very interesting candidate is TRABID. TRABID is a deobiquitinase, which essentially stabilizes Jumanji-2D, and Jumanji-2D is the enzyme responsible for the demethylation of DNA. So, in other words, if Jumanji-2D is activated, the DNA is open for the transcription of pro-inflammatory cytokines such as IL-12, IL-23, and this is just a mechanism in which the cells just might drive inflammation. We are studying the expression of TRABID in the synovial samples of patients with psoriatic arthritis, and you can see that there is a very significantly higher expression of TRABID in the synovial sample of psoriatic arthritis patients. This is correlated with the pathotype. The more inflamed the synovial tissues are, the higher expression of TRABID is. There is a correlation with the inflammatory score and a significant correlation with different immune cells infiltrating the synovial tissues of our patients, and also a very significant correlation between the expression of TRAbID, the expression of IL-23, and the expression of IL-23 receptor. So in other words, TRAbID might be responsible for the activation of innate immune, the type 3 immunity in our patients. And when we look at the cells that were expressing TRAbID, what we found was that essentially macrophages of synovial lining, sublining, were the cells expressing TRAbID, and these are the most important producer of IL-23 in our inflamed tissues. Interestingly, we also are doing collaboration with the Toronto University some functional experiments where we are trying just to see is whether just inhibition TRAbID by using this chemical compound is able to reduce the secretion of pro-inflammatory cytokines, and you can see that the different concentration in a concentration-dependent manner, the inhibitor TRAbID is able to reduce the secretion of IL-23, TNF-alpha, and IL-1 beta. This mode in this line of myeloid cells, but also in mouse splenocytes. But what is the most important information I'd like to share with you is that blocking TRAbID by using the same inhibitor is able to significantly reduce the severity of the inflammation in the SKG murine models. In black, you can see in the black line, you can see how the higher concentration or the highest concentration of TRAbID inhibitor is able to significantly reduce the severity of inflammation. And this is also confirmed at histopathological levels. So this is my last slide, and this is my last slide. And I think that we just at the beginning of our journey, we need just to join the force between derma and rheuma need to be just joined, because we need just to collaborate in a very strict way, in a very intensive way, just for explaining, for better explaining, better understanding the pathogenesis of the disease that we are continuing to see and to treat in, I think, not in a different way, but I think in a very integrated way, and this need to be absolutely just enhanced in the future. I would like to thank all the people that have been involved in these projects. I would like to thank you for your attention. I'm absolutely happy to receive your question and to answer if I can to your question. Thank you very much.
Thank you so much, Professor Ciccia, for this great talk. It is really inspiring to see also the results from the other side that basically also relate then back to psoriasis. And maybe I start with a question. So you asked if it's rather an autoimmune or autoinflammatory disease, the ankylosing spondylitis. How would you address, in general, psoriasis arthritis as an answer to the question that you asked?
This is a very, 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 very important point. And I think that me as a rheumatologist, have learned much from you, from dermatologists, and from your stratification of different forms of psoriasis in just more autoinflammatory conditions. Some form of psoriasis you learned to me that are more autoinflammatory compared to others, in which possibly adaptive immunity is more important, is more relevant. The problem is in my idea of psoriatic arthritis that some patients, especially patients with higher levels of CRP or ESR, patients with very acute onset of the disease possibly have more autoinflammatory pathogenesis of the disease and the other patients. And I think not pure autoimmune disease, because in this case, we will have autoantibodies that we just can just try in our patients to see whether or not they are positive, and just a mixed pathway, but in which adaptive immunity is more important. I have to say that we have some candidates, some autoantibodies that are candidates in our field, this anti-CD74, for instance, that have been demonstrated to really are able to very good in stratifying patients with spondyloarthritis compared to controls And CD74 is the receptor for the macrophages inhibitor factor, which is a very powerful cytokine for the innate immunity. But I think we have just to need more effort for answer, which is the most important part of the story relevant in our patients.
Question from you too, Francesco. You showed the concept of an inflammatory march that nobody knows exactly where it starts, no? So the hen and the egg. So this axis, this inflammatory march between gut and joints. Where do you localize skin in this axis?
It is a very, very, very important and relevant question. What I can tell you is that we have 50% of patients with spondyloarthritis, psoriatic arthritis that do have a subclinical gut inflammation. So in these patients, if you perform ileoconoscopy, you can find findings that are absolutely similar to the findings you can observe in patients with Crohn's disease. This is just a subset of patients in which possibly the gut is more important, more relevant in the pathogenesis of the disease. Obviously, the skin part is not possible to avoid to consider. And what I can tell you is that in our hands, when we have different models of spondyloarthritis, we can induce spondyloarthritis, for instance, by inducing skin inflammation with imiquimod, and we have, perfectly there is skin inflammation. But after several weeks, the same mice develop arthritis. They develop joint inflammation. And this is just a model that we can use for demonstrating, just for assuming that skin is important in driving inflammation in the joints. When we just compare this model with the classic model in which the gut is more important, the cardinal model of SKG mice, or actually B27 transgenic rats, what we observed is that there is a completely different phenotype of the mice. Mice in which we have induced arthritis with imiquimod do have more peripheral joints inflammation, not systemic inflammation and psoriasis, differently from patients in which we have induced inflammation starting from the gut, in which we have ileitis, skin inflammation, uveitis, axial disease, and peripheral joint disease. We have also some unpublished data in collaboration with the Erlangen group, in which we have demonstrated, the paper is under review on Nature Immunology, and we have demonstrated there is just the migration of immune cells from the skin to the joints of the different model of mice, which is the L23 minicircle mice. And this highlights the importance of the skin also in the pathogenesis of the disease. But the phenotype of the disease is different according to the site where the disease starts. I don't know if I have my answer was complete or not.
Very interesting.
Very
I think
good. Thanks
that
a lot, so there are no questions in the Q&A or in the chat function, but if you have any questions, please forward them, send them to us, and we will forward them also to Professor Ciccia. And I would like to thank again to you for your time and for this great presentation, and for
Thank you for inviting me. It was a pleasure. Thank you very much.
Yeah, and that's also a motivation to collaborate and also between disciplines. I think it was a very nice example for it. Thanks a lot.
Thank you very much.
I would now like to also emphasize the next ESDR kitchen with a new flavor. There will be the first time the ISID flavor with the theme of aging, with two big names that will present, Aline Fuchs and Ray Watanabe. So this will happen in 1 month. And then also a reminder that the webcasts are then also available on the homepage, so you can also download them after the actual sessions, and you're very welcome to do so. And just another reminder is important dates for the next ESDR annual meeting, which will take place in September in Antwerp in Belgium. So abstract submission is already possible. Deadline will be beginning of June, and the same deadline is also applicable for the travel grant applications. So please submit abstracts and also apply for travel grants for young researchers. Thank you.